Automate your antibody purification workflow at preparative and analytical scales. We have Katie SCHAEFER with BioRad and Pete WINSHIP with Teledyne CETAC. I'd like to hand it over to them for their introduction.
Good morning, everyone. I am Katie SCHAEFER. I work for BioRad Laboratories. My background is in biochemistry, and I did my graduate work studying DNA-protein interactions. So I started off my career with BioRad as a proteomics field scientist, training all of our users in the Southwest United States how to use our proteomics equipment. So I have transitioned and now am the global product manager for the NGC chromatography system and our accompanying ChromeLab software. So just a little bit about BioRad. If you don't know about BioRad, it was started in 1952. It is a husband and wife team, David and Alice Schwartz, who started the company. And you can see this picture on the right that I love so much. This is their first office, a Quonset hut in Berkeley, California. So their goal was to create a company that could help advance the scientific industry and advance scientific discovery. This company went public in 1966, and we are a global leader in clinical diagnostics as well as life science research. The headquarters today is located in the East Bay of San Francisco in a town called Hercules, California.
Hello everyone, my name is Pete WINSHIP. I'm a technical product manager at Teledyne CETAC Technologies. My background is in chemistry, particularly analytical chemistry. I undertook my undergraduate studies in applied chemistry and then postgraduate studies in analytical chemistry with a particular view on trace element analysis with ICP-MS and ICP-OES instrumentation. Following that, I spent 5 years working for the Medical Research Council in the United Kingdom, and then I joined what was at that point CETAC Technologies in 2011. So just some background on Teledyne CETAC Technologies. So the company was founded, it's a slightly younger company. It was founded in 1985 in Iowa in the USA. And the company started its life manufacturing the U5000 ultrasonic nebulizer, which is a system that we still manufacture and sell today. In the 90s, we introduced the first auto sampler, and this started the foundation for what the company is today in terms of automation. It was sold to Steve Dwyer in 2000, one of the original stockholders, and then acquired by Teledyne Technologies in 2013. And today the company manufactures automation, sample handling and production systems in its main facility in Omaha, Nebraska.
All right, with that, we're going to jump in and start talking about the meat of our presentation today. And that's going to be specifically protein purification revolved around antibody purification and how you can automate your workflow using the NGC system as well as our auto samplers from CETAC. So a brief introduction into protein purification. Why do you care? The reason is that every single biochemical and biophysical researcher needs to make pure protein to do their downstream experiments. And I'm sure, as you're aware, there are now biological drugs that are on the market. And all of these biological drugs, the research and development of them are going to involve the purification of very specific proteins as well. So the goal of this is you're going to have to overexpress that protein of interest in some sort of cellular machine like E. coli, yeast, or other cell cultures. Once you overexpress it, you have to then selectively isolate that one protein that you care about from the rest of all of the other biological material that a cell can hold. So it's a pretty daunting task. It's a whole kind of kitchen sink event, and you have to pull out one thing. So not only do you have to pull out your one protein of interest from an entire cell's worth of material, you need to purify large quantities of high purity so that you can use these proteins effectively in your downstream experiments. And ultimately, our goal number 3 would be, if you are going to use them for therapeutic treatment, they need to be extremely pure so that you don't cause any detrimental effects to the person that you inject them into. So protein purification is typically done through chromatography, medium pressure to low pressure liquid chromatography systems. And it is not a one and done process. If you look at the right, this is the typical workflow of what it can take to get a pure enough protein for your needs. Typically, it will begin with some sort of capture step. So we will capture our protein of interest, and we could do that if it's a tagged protein where we engineered a tag onto it. So like a 6-histidine tag, we can use that to specifically interact with a IMAC column. Or in the case of what we're going to talk about today, we want to isolate a native protein, a protein that has no engineered tag on it. And so we can use a property. So in the case of IgG antibodies, we can use a protein A column, which binds tightly to the IgG to isolate all of the IgGs out of that solution. So it would be wonderful if your protein was pure enough after that first capture step, but maybe it's not. So then you'll have to do another run of chromatography that we're going to call an intermediate purification. These intermediate purifications can be something like ion exchange or desalting or hydrophobic interaction. And this typically does a wonderful job at getting you to higher purity, but sometimes that's even not pure enough. And you'll have to move forward to a third round of chromatography, which is then called polishing. And in this polishing round, it would be a column that is a resolute quality resin, maybe some size exclusion chromatography, or a very high quality mixed mode. So you can see with each round of purification, you increase your purity, but you also increase your procedure time and increase the amount of hands-on interaction that's needed. Not only that, at the end of it, you definitely need to confirm your purity as well. So you can confirm your purity through standard gel electrophoresis techniques, or you can confirm your purity through analytical chromatography as well. So when we talk about protein chromatography, there's pretty much 2 different types. There's preparative, so creating small to large scale batches of the protein of interest that you're trying to produce. Or there's analytical, where you're doing very small aliquots of the protein that you made in the preparative run to do high resolution separation to get confirmation of purity. All right, so today we're going to talk about our systems being automated with autosamplers with respect to purifying monoclonal IgG antibodies. So IgG antibodies are the most prevalent in your body. So typically in your blood and your plasma, you have about 75% of your antibodies are IgGs. So they are highly specific biomolecules that come from the immune response to some sort of foreign object, some sort of antigen that is introduced to your body. So this is basically how your immune system will recognize it they will have an antibody ready to bind it, and it'll be essentially a beacon for the immune system to come in and remove this foreign object from your body. The reason why we love to use them in biochemistry and biological research is that specificity is fantastic. And so antibodies have been used and created and are standard in many types of experiments, such as Western blotting, ELISA, and flow cytometry. These monoclonal antibodies are typically produced in a couple different ways. So you can do hybridoma cell lines, you can do phage display technologies, or single sorted human B cells. Today, we're going to hypothetically talk about doing a hybridoma cell line. So not only are these wonderful for in vitro laboratory experiments, but their specificity also makes them a very exciting target for drug candidates. So the world of biotherapeutics has been creating some antibodies, and actually there's a bunch that are out on the market today, and if you're in the US, you've probably seen some commercials for them, that they target very specific antigens within your body that are representative of a disease state. So basically, it will elicit your immune response, targeting it to the appropriate locations. So some that you may have heard of, Humira and Rituxan. Humira is a treatment for arthritis, Crohn's disease, as well as ulcerative colitis. And Rituxan is a treatment for certain autoimmune diseases, as well as cancers. And you can see in parentheses are their official names. And these antibodies both end with MAB, and that stands for monoclonal antibody, an antibody that is specific against one antigen. All right, so let's say we're going to go ahead and develop a new antibody, and we have our target in mind that we want, and we need to first screen a whole bunch of antibody candidates for efficacy testing to make sure we could choose which antibody we should move forward with for development. That is going to be screening of antibody candidates on a larger scale. The second thing we're going to talk about is confirming the purity of this production, so of the antibody candidates we made in the first step, with a smaller scale, more analytical workflow. All right, so before we dive into the details, let me tell you a little bit about our NGC chromatography system. Honestly, this is one of my favorite toys. It is a medium pressured chromatography system that is very flexible and customizable to meet your research needs. So the image toward the center of the screen, if you can see my mouse, is the NGC Quest. And that's one of our more basic systems where you have at the very bottom 2 reciprocating piston pumps that work together as a team to create very precise gradients for doing elutions. You also have up here your inlet valve, your injection valve, where all of your samples get introduced. And then over here, you will have a detector. And so you have your choice of a single wavelength detector or a multi-wavelength detector. So our systems are all run with what we call our ChromeLab software, which is wonderful. It is very intuitive. And the wonderful thing also about it is that all of our systems, no matter as basic or as complex, like you see over here with the NGC Discover Pro, will all be run off of the same software platform. So the NGC Discover Pro on the right-hand side is an example of one of our pre-configured systems that is highly customized. The additional things on this system include buffer inlet valves to automate the cleaning and automate multiple column runs. Column switching valves that will allow you to have multiple columns permanently plumbed on your system. A sample inlet valve to pull in multiple samples onto your system. A sample pump specifically for introducing large amounts of sample to your columns. In the center with the green, that's our buffer blending valve, if you are interested in having the system make your buffers for you. And we also have a pH monitor and an outlet valve to give you a lot of versatility. Not even that, you can see here on this NGC Discover Pro, there are 3 open bays. You could even add more functionality with more column switching valves if you desired. All right, so it is very flexible. It grows with your research needs. And we also have flexible fraction collection options as well that we'll talk about a little bit later. All right, Pete.
Yeah. Thank you, Katie. So what automation do we have to automate the protein purification workflows, and why would you want to automate these workflows? Well, this comes down to actually differences in the sample volumes that are available for each of those workflows. So what we've got here is we've got 2 types of automation which meet the requirements for those two ranges of sample volume. So we've got the ASX 500 series automation. So this is the an example of which you can see in the autosampler at the bottom of those images there. And this automation is designed for operating with milliliter sample volumes and that range of sample volumes. And we've got the MVX 7100 microliter workstation, which is specifically designed for handling microliter sample volumes, a microliter range. So the 500 series is predominantly used for sample introduction for the preparative protein purification workflow, whilst the MVX 7100 is used for the analytical protein purification workflow. So both are robust and reliable autosamplers. They're both configurable to your analytical requirements. Now, why would you use these? Well, you can put both of these in place with the NGC system to improve the efficiency of your protein purification processes. So you can set these up so that you can have unattended operation with your NGC system. So you can have them running 24 hours a day and 7 days a week. And this would allow you to do, for example, the purification of 84 antibody candidates over a period of 2 days. So it will offer you accuracy and precision for sample introduction processes that you want and a high level of reproducibility.
All right, let's talk about preparative protein purification and what this workflow would look like. So like I mentioned before, what we're going to do in this case is screen our antibody candidates. So we will express our multiple IgG antibody candidates in a hybridoma cell line on a small enough scale that we're able to make enough protein that would allow for our efficacy testing, but also on a scale that allows us to efficiently purify many different candidates. So in this case, we're going to use the ASX 500 autosampler with an NGC system that has a sample pump, inlet valve, and fraction collector. This type of preparative purification is going to include a capture chromatography step as well as an intermediate chromatography step. So we're going to do protein A and then desalting. So what is this workflow going to look like? We are going to first prepare our cells and culture them into flasks, and then take that culture media and transfer it into conical tubes that will then get put onto the ASX 500 series. Once the tubes are in place, we'll set up a batch on the ASX. We will set up a series of methods on the NGC, which will allow the systems to communicate together and our samples will be taken from the ASX automatically and loaded directly to our first column on our NGC instrument. The method that I'm going to be discussing is a tandem chromatography method. So tandem means it's going to be a 2-column method where we are going to run across both columns before the sample is eluted and collected into the fraction collector. And so this is going to give us really high automation and pure protein. So our fractions get collected in whatever sizes we desire. And then, because we've already set up this series, it's going to repeat by itself as many times as the autosampler will allow. The data that gets generated, we can observe this within the ChromeLab software. We can look at each run individually, or we can look at overlays of the entire series together. From there, we can confirm the purity of our samples, either through standard gel electrophoresis techniques.
Katie, are you there? Seems to have lost
here.
your audio.
Can you hear me? I'm here. Can you hear me? Hello?
I can hear you, Katie.
Okay, I will continue. Sorry about that. So we'll confirm our purity through gel electrophoresis, or you will confirm our purity through our analytical separation. So our antibody candidates, we're going to culture them up in culture flasks, and these hybridoma cells, they could be adherent, which is very lovely, because what's going to happen is that they're going to excrete their antibodies into the media of the culture. So then you can decant that media and transfer it into your conical tube, which then gets placed onto the ASX autosampler, which right next to it, you can see the NGC system and our NGC FC fraction collector.
Thank you, Katie. So starting with our sample tubes, we're going to then put them onto the 500 series autosampler. So we've got 2 types of this autosampler. There is what we have here is the ASX 280 autosampler, so the image there on the top right-hand side, and then the ASX 560 autosampler. So these are autosamplers from the same platform, and they only differ in terms of the sample rack capacity that they have on them. So the ASX 280 is a 2-rack autosampler, and the ASX 560 is a 4-rack autosampler. So these are both robust, reliable XYZ autosamplers, which are designed for handling sample volumes in the tens and hundreds of milliliter level. And there's a number of different options of sample tubes and bottles to work with for this system. So this system is resistant to the type of reagents that you'll be working with for protein purification. And it also is configured for a number of rinsing actions between sample introduction processes. So how does the 500 series automation interface with the NGC system? Well, I'll start off here with this schematic on the left-hand side here. So both the NGC instrument and the 500 series automation, they share a common operating PC. So you'll see there that laptop between the two. So both systems are connected to that operating PC. And then there's another communication cable between the automation, the 500 series automation, and the signal import module of the NGC system. And then the automation itself is operated with a piece of software that we have called AScript, and that's how we get the communication signals between the two. So a sample introduction process, how does this work? Well, everything starts with the NGC instrument. So you have an operating method on the NGC system, and you start off an analytical batch. And the first things that happen are you have a number of column equilibration and baseline zeroing processes. Now, when the NGC system is ready to proceed, it will send a communication signal to the automation, at which point the sample probe of the autosampler will move to the sample position that's required, and then a pump on the NGC system will pull the sample up through the sample probe and through the line that's connected to the sample inlet valve via an air sensor. So when all of the sample has been pulled up to the inlet valve, the air sensor will recognize that there's no more sample to come up, and the pump will stop pulling the sample through, and the sample will then be separated on the column, and the system will go through its analytical process. At that point, the automation will go through its rinse cycle and get itself ready for operating again for the next introduction of the next sample.
So at that point, the ASX is washing, but our protein sample, our IgG, has already been loaded across our first column, the protein A column. So if I walk you through this fluidic scheme, this circle up here is our sample inlet valve to which the ASX is connected, as well as other buffers and cleaning solutions. That connects down to our sample pump. And as the sample is being loaded from the ASX, it is being driven from the sample pump to a protein A column that is on our first column switching valve. So our first column switching valve will be online, and our second column switching valve will be offline. So all of our protein sample will be run across the protein A column. Once it's gone across the protein A column, our IgG protein that we want to capture in red, hopefully all gets captured on there. And then we'll do a wash. Once we wash out the remainder of any unbound protein, we will then do an elution. So the typical elution for a protein A column is a really low pH solution, pH 3.0, which is not favorable for the majority of antibodies to be sitting in for a long time. So what we're going to do as part of speeding up our purification and making sure our samples are going to elute in a condition that is more stable for them, when we elute off of the protein A column, we are going to bring the desalting column on the second column switching valve online. So whatever comes off of the protein A column will get directly loaded onto the desalting column. The desalting column will have already been equilibrated in the buffer that we want our protein to end up in. So in this case, a buffer at pH 7.3. Desalting column separates based off of size. So larger molecules like proteins and antibodies will travel much faster through the column than smaller molecules, like all of the components in the buffer that eluted our antibody off from the protein A. So what we have is now all of the pH 3.0 buffer components are separated away from our protein of interest, our IgG, and our IgG then elutes off and fractions are collected in that favorable pH 7.3. So what does this method look like? I tried to make this screen look as much like our method as possible in the software. So it would begin with equilibrations of your first columns. So your desalting column and your protein A column. And then the baseline would be zeroed, making sure everything is ready and zeroed prior to injection. After that, this sample application phase is reached, and it sends a trigger to the ASX. The ASX will begin its process, and the time zero down here is the beginning of our injection. And you'll see as the ASX is loading the sample, we see our absorbance of 280 rise. and we reach our end of our sample, and the loading stops, and our purification moves forward based off of the air sensor. So the air sensor is set to, within the software, as end of sample, so it knows when it reaches that end of sample to move on with the rest of the method. The rest of the method is then washing the unbound protein out of our protein A column. So we have this wash right here, and we see we kicked out something. And then we're going to jump up this black line to 100% B, which is our elution buffer for the protein A column. And we're going to, at the end of it, bring our desalting column online so that all of that protein coming off of the protein A column goes directly to the desalting column. We then continue running just the desalting column with the desalting buffer. And here we will be collecting fractions of this entire phase. And what we will observe is a nice, beautiful peak of our pure IgG protein at pH 7.3, followed a little bit later by this peak in conductivity, which represents all of the other buffer components that we separated it out from. After that, we have two more steps that are going to be doing cleaning and regeneration of the columns, and the entire flow path of the system will be flushed so that it is ready to be introduced to the next sample that the ASX has ready for it. And we'll repeat this as many times as we like. So like Pete mentioned before, if you're using 50 milliliter Falcon tubes, that means you can do this 84 times. And with this method, that will run for about 2 whole days. So you can set it up and let it run over the weekend. Here's some data from some real IgG purifications. On the left, you can see 18 traces that were run automatically without somebody interfering. And you can see how beautifully they all overlay. The customer who did this is a quote from them. They say, the method is easy to use and truly hands-off. And on the right, let's take a look at just a different view. We have 8 of these traces, and they're stacked on top of each other. And I think the most important thing is that not only is this reproducible and reliable, but it ensures that all of your samples are eluting in the same exact conductivity and the same exact pH every single time. And so having this automation with the auto sampler and the NGC really allows for you from day to day to user to minimize the amount of variability in your sample preparation. So how are we collecting our fractions? So right here, I'm showing you our NGC fraction collector, which is wonderful for high throughput applications. You have your choice of up to 4 different types of bottles or racks to collect into, which means you can mix and match the types of bottles and racks that you want to use in any given method. And if you're extremely high throughput, you can actually attach 2 of these fraction collectors to your one NGC system. It supports flow rates up to 200 mils per minute. We have Peltier cooling options as well, or it is sized perfectly so it will fit into a deli fridge. The system not only will collect based off of windows in the Chrome Lab software, but you can also use triggers such as slope of your absorbance, pH, and conductivity. So this is a wonderful option that is great for your high throughput needs. One of the concerns with running things automated and running different candidates is that there might be cross-contamination from one run to the next. And also doing this automated purification, the concern might be maybe the purity isn't as good as you would like. But here, I want to give you an example of this customer who did these purifications, starting with 50 milliliters of sample. They were able to recover about 2 to 6 milligrams of IgG per run. The purity, based off of gel shift, is greater than 95%. And they confirmed through LCMS that there was no cross-contamination from one antibody species to the next. All right, Pete.
Thank you, Katie. What we've just talked through is basically, just to conclude this section, we've just talked through an automated screening solution for the preparative protein purification steps. So we've shown that you can automate the introduction process for the separation, and then you can collect the fractions that you want. So this can allow you to undertake separation of, say, up to 84 antibody candidates over the course of 2 days, so a weekend. And it's very easy to implement to do this, and a number of different phases of and dimensions of chromatography can be applied as part of this. So next, so we're going to leave the preparative protein purification behind and move on to the analytical separations and purification workflow.
All right, with the analytical purification, we're going to be talking about this in terms of confirming the purity of the production that we just did in our screening. So we're going to do an analytical analysis using the MVX microliter workstation in conjunction with an NGC system that has an analytical flow cell to ensure that we get nice sensitivity with the small amount of protein that we are going to be injecting. Typically, these types of purifications are going to be done on a polishing type column, so a size exclusion column or a really high resolution resin column. So this will give you purity. You will know if your sample is going to be good to go. Sometimes people also will do maybe fluorescent conjugates to their antibodies. And this is additionally another way to confirm that that reaction worked appropriately. So the workflow for this, again, will be take your samples, prepare them how you need. So take maybe just an aliquot of the antibody that we made in the step before, or if you're doing some sort of conjugation to it, do your chemical reaction, and then take an aliquot of your new conjugated antibody. Then transfer it to the MVX workstation, and these can be in 96 well plates or bottles, and the wonderful thing is that they can be covered. Then the sample uptake, again, we're going to be setting up a batch on the MVX and setting up a series of methods on the NGC and start them together. So the sample will be taken up from the MVX and introduced to the NGC, this time through the injection valve, through the sample loop position. Then we'll go on to do our high resolution purification. If you want, you can do small scale fractionation, but typically a lot of the times here, the data that you're going to derive from this run is the information that you need, and you don't need to collect anything. And then, just like before, we're going to repeat it as many times as you have samples on that MVX workstation, or as many samples that you need to do that day. From there, the data that gets generated, you can look at it individually, or again, you can overlay these traces very easily within the ChromeLab software. And this data is your purity confirmation, or maybe for some other experiments, maybe doing different protein binding kinetic experiments, you might want to do some other downstream purification or downstream confirmations as well.
Thank you, Katie. So at the start of this, we're going to be talking about essentially sample volumes. So for the analytical workflow for protein purification, you will be working with sample volumes which are considerably lower than you were working with for the preparative workflow. So for this, we're going to use the MVX 7100 microliter workstation, which is the system that you can see here on the right. What we have here, I'll just quickly go through this, is that we've got a main section of automation, so XYZ movement, and then on the right hand side of that you can see that there's an additional module, and that's comprised of a short syringe pump module there at the bottom, syringe pump there, and then above that there is a 6-port switching valve onto which there will be a sample loop that we're going to pull our sample into. So this is accurate and precise XYZ automation. It's been designed to handle samples in the microliter range, so down to around about the 5 microliter mark up to 950 microliters. And from this automation, you can sample from a number of different rack and well plate options. So, for example, you can work with the VT54 sample racks. So these are HPLC style sample vials with septum pierceable caps. So you can have a small volume of a sample in such vials and can have them sealed and keep them protected from the outside environment, so you can minimize evaporation and contamination. You can sample from 96 well plates, a number of different types, anything up to 44 millimeter in height. So you can sample from the lower capacity 96 well plates, where you might have about a capacity of about 300 microliters per well, up to the high capacity 96 well plates that have a well capacity of about 2 milliliters. And then if you can also work from 384 well plates as well. And you can get 2 racks or well plates on the front of this system. We also have very fine control of the sample uptake process. So for larger sample volumes within this scale, where you might have a couple of 100 microliters of sample that you want to load and introduce to your NGC system, you can pull that relatively quickly. But if you've got lower volumes, say 10, 20 microliters or less, then you can slow down the uptake process so that you've got very fine control and accurate and precise sample uptake, so you can get consistent injection volumes with very low volumes. In addition to that, there's options for temperature control, so you could cool your samples down to 4 degrees centigrade. So if you have concerns about evaporation, or if you've got a particular sample with a volatile component, then you can cool down your sample vials or well plates to protect them from evaporation. Also, you can heat up your samples as well, up to 40 degrees C if you want, if you required some sort of incubation temperature for your samples. And then, like the ASX 500 series automation, this system is resistant to the type of reagents that you will be using for this protein purification work. So just quickly before I move on, just a quick description of how this system works. So this is slightly different to the 500 series automation in how we introduce a sample. So in this case, the sample probe of the system will move to a sample position, and the syringe pump on this system will pull the sample up onto the sample loop that's on that 6-port switching valve. And then the sample, when it's ready on that sample loop, that sample is going to be switched into the mobile phase flow that's going to your separation column on the NGC system. So how does the MVX 7100 system interface with the NGC system? Well, it's quite similar to the ASX 500 series system, actually. So like before, you have a common operating PC, which you can see here in the schematic on the left. So the NGC instrument and the MVX 7100 microliter workstation share that common operating PC, and both are connected to that. And then there's a communication cable between the automation, the MVX 7100, and the signal import module of the NGC instrument for communication purposes and telling the automation what to do. And much like the 500 series, we have a piece of software for control of the automation, and in this case it's called CETAC WORKSTATION. So the sample uptake process, so similar to operation with the 500 series automation, everything starts with the NGC instrument. So you start an analytical batch with the NGC system, and the first thing that happens is that the separation column will go through an equilibration phase, and then the baseline for measurement will be zeroed. So when the NGC system is ready to proceed, a communication signal will be sent to the MVX 7100 microliter workstation, and the sample probe will move to a sample position that you're interested in. The syringe pump, as I mentioned before, will pull the sample onto the sample loop, at which point, when the sample's in the position on the loop, the 6-port switching valve will switch the sample into the mobile phase flow that's going towards the separation column. At which point the MVX 7100 will go through its rinsing cycle and will clean itself and back flush, ready for the next sample introduction of your next sample. So before we move on, I wanted to say something more about the CETAC workstation operating software. It's a very flexible piece of operating software for this automation. So what this will do is, it will, just to go through this quickly, you will use this for selecting the sample locations, for selecting the sample injection volumes that you're interested in. So like I said, that can be anything down to 5 microliters up to 950 microliters. And then you can use this here to change the uptake flow rates. So like I said, for small volumes, you're more likely to slow it down. For those larger volumes within this range, you could make it move faster. You would also use it for controlling the temperature of the samples between that temperature control range, and configure the rinsing processes as well. Now you could also use this for different injection methods. So there's 2 approaches to the injection process here, and they're based on the kind of sample volume that you have available. So what we have here is we have a full sample loop injection approach, and we've got a partial loop injection approach. So you would decide which one you want to use based on the kind of sample volume that you want to work with. So with the MVX 7100 system, we provide 2 sample loops. We provide a 100 microliter loop and a 500 microliter loop. And what you could do with either of these loops is that you use the CETAC workstation software to tell the system to fill the sample loop with a slight excess of sample, and then you can switch that whole sample loop volume into the mobile phase flow to your NGC. So in this case, you'd choose a slight excess of sample, and you could switch in 100 microliters or 500 microliters of sample into that introduction flow. But what we have is we have the ability to completely change that sample loop. It's very easy to do. A sample loop is just a length of tubing that's connected to the 6-port valve. And just to give you an example, at a customer site recently, we set up a system where we used a 25 microliter sample loop. And we set up a method to completely fill that sample loop with a slight excess of sample. So we used 27 microliters, filled the loop completely, and then when that 25 microliters was switched into the introduction flow, we then sent the remaining 2 microliters, so 1 microliter either side of the loop, to waste. So we were only wasting about 2 microliters. So so that works very, very nicely for larger sample volumes down to a certain point. Now, if you've got a very low sample volume, so say maybe 10 to 15 microliters or less, you might want to use a different approach. So in this case, you can use this partial sample loop injection method. So here you would put on a relatively large sample loop, so say 500 microliter sample loop on the 6-port switching valve, and you can instruct the system to pull a small volume into the middle of that loop. So say 10 microliters, for example, into the middle of the 500 microliter loop. And on either side of the sample, you would have buffer solution. So you're filling the loop completely with liquid liquid, but only 10 microliters of it is sample, the rest is buffer. So there'll be no air being introduced into your mobile phase flow to your NGC system. So we switch in that whole volume of liquid, buffer, sample and buffer, and then we introduce our low volume that way. So that's the alternative way of operating this, and it's particularly applicable to those low volumes.
So I have been able to generate some data for both of these types of sample loading. So what I'm showing you here is an overlay of 48 runs of full sample loop uptake and injection. And this was a protein standard that I was injecting without a column, just straight on the system to see the reproducibility. I actually was able to do a whole 96 well plate, but I'm just going to show you 48 here. So this was a 25 microliter full loop injection, and you can see how consistent these injections were from one sample to the next. And this machine ran beautifully over 96 samples, which took about 18 hours. But I think more exciting is this. This is the MVX using the 10 microliter partial sample loop uptake. And what I've done here is I used BioRad's gel filtration standard, which is commonly used in the research world. And on the bottom left, you can see an overlay of 5 traces. So this is 10 microliters of the gel filtration standard at the same concentration run over our ENRICH 650 size exclusion column. And you can see how beautifully those 5 different injections overlay, how beautifully reproducible these injections are. And if you look at the right-hand side, you can see in the ChromeLab software, we have a very nice feature where we can split out these overlays, and you can see how nicely these systems perform together.
Lovely. Thank you, Katie. So at this point, we'd just like to conclude and wrap up. So we have a quick conclusion slide, just to have some concluding comments. From Teledyne CETAC Technologies, we can provide the 2 types of automation that I described to easily streamline the protein purification process with the NGC instrument. So these systems will allow you 24-hour-a-day operation or automated unattended operation with the NGC system 7 days per week. So it will allow you to have accurate and precise sample introduction, which is highly reproducible, and it will allow you to meet the full sample volume range requirements for protein purification workflows.
Thanks, Pete. And so with the BioRad NGC, it really is an intuitive system that can adapt and grow to meet your most basic to most advanced purification needs. With the auto samplers from CETAC, you really truly can automate your purification at both preparative and analytical scales. And I think most importantly, have the consistency and reproducibility that you would need day to day and different operator to different operator. And with that, we'd like to thank you for your attention.
Thank you very much for joining us. Thank you. And please feel free to contact either Katie or myself at the email addresses below. Feel free to access the BioRad website or the Teledyne CETAC website to have a look at the systems that we've been talking about today. Thank you very much.
All right, well, we'd like to open it up to questions. So if anybody has questions, please feel free to type them in. We received several questions. We only have a few minutes left of time, so we'll go ahead and take a few, and then those that we don't get to, we will follow up individually at the end of the webinar. So I'll start with a couple of these questions. First one that has come in: how can I monitor an overnight run to ensure there are no problems?
I'll take that. That's a great question. So the NGC system, you can network it, and you can network your computer, and you can remotely monitor what's going on with your system through having it send you email updates. You can have it a VNC app on your phone or your iPad, and essentially have the touchscreen control. But my favorite is that you can use a software like TeamViewer and take over control of that computer wherever you're at, and have full control and see everything that's happening with the system. So you can truly even just walk away and check in without physically having to go there. Just to let you know, I also do have a customer that starts their system in the morning before they get to work and gets there to perfectly equilibrated columns. It's wonderful.
Okay, thank you. Next question. What size of sample loops can be used on the MVX 7100 system?
Thank you. I'll take that one. So we provide two types of loop, like I mentioned, 100 microliter capacity or a 500 microliter capacity. Really, it's the sample loops are a certain length of tubing with a certain internal diameter, and they are fitted with fittings and ferrules onto the 6-port valve. So we're not restricted to using just the 100 or the 500. We can make we can work with a sample loop of any different size, really. From like I said, the example I gave was a 25 microliter loop that we established as a customer site. You can have loops in the milliliter size if you were so inclined. And the CETAC workstation operating software is flexible enough to allow you to put on any different size sample loop that you want to work with. So if you've got a particular sample volume in mind that you'd like to do the full sample loop injection for, then that's something that we can talk about setting up.
So I think this is also for you, Pete. What is the maximum volume of sample that can be introduced with the ASX 560 auto sampler?
Maximum volume, so typically you'd probably work with 50 ml sample tubes, but there is another option for the ASX 560, which is 250 milliliter sample bottles. So that's the maximum volume that you'd be able to work with in a sample position, would be that size of bottle.
All right, it looks like we can probably fit one more question in to respect everyone's time. This one is, how do I avoid running out of buffer introducing air?
I think that's for the NGC. Great question. So we mentioned in the programming that we have an air sensor to detect end of sample. You can actually have up to 8 air sensors per NGC system, and that's going to allow you to sense either end of sample, where the programming knows to move forward with the purification, or you can set them to be end of buffer. And so I would recommend having at least one air sensor for each pump. So for the sample pump, it will be sending end of sample, and for the system pumps, it will be sensing end of buffer, so that if you do run low, it will pause your programming until you can come in and get rid of that air bubble and get your new fresh stock of buffer back on your system.